Hi, I'm Christabella. The paper I reviewed about is the Phylogeny and Biogeography of Ficedula flycathers. The novel results from fresh source material. Genus Ficedula. This genus has been model system for studying speciation, genomics, biogeography, and evolution of migratory behavior. However, no multilocus molecular phylogenetic hypothesis exists for this genus. Hence, the current research of phylogenetic hypothesis resulted in a simpler biogeographic scenario with fewer transitions between regions and fewer transitions between seasonally migratory and resident character states. Introduction of this research. The Old World flycatchers in genus Ficedula constitute the largest genus in this family taxon. This genus is ideal model in this taxon because it occupies much Old World and exhibit a variety of life history characteristics. Also, it shows variation in many migratory behavior. Last, they span many biogeographic realms. Methods applied in this research are: first, taxon sampling and sequence. All the DNA sequences derived from frozen tissue. Second, phylogenetics analysis. This research is focused in the geographic differentiation of Ficedula species. Samples of all species, most subspecies and population of resident Philippines, Ficedula and congeners across Southeast Asia. In this phylogeny, geographic distribution, migratory behavior, and current taxonomy were recognized. Biogeography and migratory behavior. Clade A reconstructed to have an ancestral range of Eastern Palearctic. Clade D, ancestral range of Wallacea. Clade C, 3 possible ancestral ranges but included the Western Palearctic with or without the Eastern Palearctic and Himalayas. Clade B, 4 possible ancestral ranges, but they consisted of combinations of Himalayas, Southern Asia, and Eastern Palearctic. New phylogenetic hypothesis resulted. All sedentary Ficedula sp constituted a well-supported clade, which was the same clade that included all Philippines and Wallacea. The analysis indicated that the most recent common ancestor of Ficedula was likely a long-distance migrant. Phylogeny and taxonomy. The genus was strongly supported as monophyletic. All samples from Philippines form a clade that was embedded inside clade C, whereas all the other samples form a clade distantly related to Philippines birds in clade B. There is a similarities and some differences between the two clades of birds from Philippines and birds from non-Philippines. As you can see in the table, there is similarities and differences displayed. At the end of the research, there are supported phylogeny of Ficedula. The phylogeny supported an origin of Ficedula are Eastern Palearctic, whereas other analysis supported an origin in Southeast Asia. Long-distance dispersal events between regions were rare in this phylogeny research. Early colonization of distant islands of Wallacea and Philippines could have resulted from a loss of migratory behavior in wintering population. After attaining a broad distribution across Eurasia, subsequent diversification largely occurred within biogeographic regions. Few discussions were made in phylogeny and biogeography of genus Ficedula. Single gene analysis revealed many anomalies among published sequence data. Other discrepancies among DNA sequences appeared to result from contamination of heterogeneous DNA sources. The result of this research add the evidence that uncritical use of suboptimal source material can lead to sequencing errors and mislead phylogenetic interference. Thank you.